Okay, taking a look at nucleic acids and protein synthesis. Just some things to think of as we go through this chapter. Where is DNA found within the cells? Why is DNA sometimes called the molecule of life? It can be argued that the discovery of the structure of DNA is one of the most important discoveries ever made by humans. How would you justify this statement? This is an interesting article, editing humans, editing the genes to correct the faulty DNA that leads to diseases. If you would please pause the video and take a quick moment to read through the article. The molecule of life. Genetic material for living things needs two features: the ability to store information, in other words, instructions for controlling the behavior of cells, and two, the ability to copy itself accurately. When cells divide, the daughter cells must have exact copies of the genetic molecules. It was once thought that this molecule was a protein. However, in the 1940s to 1950s, evidence came about that the genetic molecule was DNA. DNA must be able to carry out instructions, construction and behavior of cells, can be copied perfectly. The nucleus must pass on the exact copy of the genetic material to the daughter cells. Taking a look at DNA and RNA structure. Deoxyribonucleic acid, which is DNA, remember, has the double strand. And then the ribonucleic acid, which is RNA, is a single strand. The polymers are nucleotides, and since both of these structures have multiple nucleotides, that would be polynucleotides. Your nucleotide, remember, would be the phosphate sugar and then the nitrogen base. Nucleotides are made up of a nitrogen-containing base, a pentose sugar, which could be deoxyribose for DNA or ribose sugar in RNA, and a phosphate group. You might recall in 9th grade biology, we had the sun, the house, and then the flag, the phosphate group, the sugar, and then the nitrogen base. Looking at an actual example, here's your phosphate group, the sugar, and then the nitrogen base. The phosphate group gives the nucleic acids their acid nature. The nitrogen-containing bases found in the nucleotides: adenine, thymine, which is only found in DNA, guanine, cytosine, and then uracil, which is only found in RNA. Notice when you look at their structures, the adenine and guanine have two rings. They're known as the purines. The pyrimidines are only one ring. Looking at the polynucleotides, they alternate sugar and phosphate, and they're linked together with the bases projecting sideways. They're referring to, you have your sugar, phosphate, sugar, phosphate, and then your nucleotides are going to project sideways. They'll come out here. This is what's going to lead to what we call the ladder produce that double helix. The DNA molecules are made from two polynucleotide strands lying parallel to each other on opposite directions. So this would represent one. This would represent the second one. You would have another set on this side. The strands again are held together by hydrogen bonds, meaning our nitrogen bases are held by hydrogen bonds. The purines, these are adenine and guanine, and you can use this little saying, pure as gold, meaning pure, like purine, as gold. The A stands for the adenine, and then the G in gold is the guanine. The pyrimidine bases are going to be cytosine, thymine, as well as uracil. That's where the saying cut, meaning your cytosine, uracil, and thymine. Remember, purines are going to have two rings. If you look down, you see there's one ring here and then a second ring, one ring, and then a second ring. The complementary base pairs, A is always going to pair with T in DNA. If you're looking at RNA, A is going to pair with U. G is always going to pair with C. In 9th grade biology, you were allowed to use A, T, U, G, and C, but now you guys need to make sure that you're actually using the names. So adenine is going to pair with thymine. If it's RNA, adenine is going to pair with uracil. Guanine is going to pair with cytosine. This is just another diagram to give you the idea for the structure. Again, you have your phosphate, sugar, phosphate, sugar. You've got your nitrogen bases. Notice with cytosine and guanine, there are 3 hydrogen bonds. Between thymine and adenine, there's only 2 hydrogen bonds. Also know that this is anti-parallel, meaning notice how this goes phosphate, sugar, but then the other side is going to go sugar, phosphate, sugar, phosphate. Notice your sugar has been inverted. So that's what they call anti-parallel. DNA is referred to as a double helix, right-handed. RNA is a single polynucleotide strand. What do we mean by right-handed and left-handed? If you hold your hand in this direction and make circles, if it's moving in a circle and it's moving towards you, then that's going to indicate that you're making right-handed DNA. If you move it in the opposite direction and it's moving away from you, that's going to indicate that you're looking at a left-handed DNA. These models are mirror images and they cannot be converted from one into the other by rotation. The helix of normal DNA is right-handed. Left-handed helixes have been produced experimentally and may be present in living cells. All right, quick question for you. If you would pause the video and take a moment to answer the question. The structure of ATP. ATP, which is known as adenosine triphosphate, is not part of DNA or RNA, but it is a nucleotide. The structure of ATP has three components: adenine, ribose, and phosphate. The adenosine can be combined with 1, 2, or 3 phosphate groups. AMP, which is the adenosine monophosphate, or ADP, adenosine diphosphate, or ATP, adenosine triphosphate. So the prefix would change depending on the number of phosphate groups you have. The adenosine is going to form from the adenine and the ribose combining together. Be careful. Do not confuse adenine with adenosine. Adenosine, again, is adenine with the sugar. And don't confuse thymine with thalamine, which is a vitamin. Two nucleotides can join together by a condensation reaction. Here's one nucleotide and a second nucleotide. Two nucleotides form a dinucleotide, di meaning there's two of them. The bond formed is a phosphodiester bond. It's a diester bond because the phosphate group has two ester bonds, one at each of the sugars. You could have several nucleotides bonded together. You could have several million of them bonded. That's going to make a polynucleotide. The phosphates and sugars are all linked by phosphodiester bonds to make the backbone. The bases are going to be at right angles to the backbone. All right, question time again. If you would please answer the question before moving on. By the 1950s, the structure of polynucleotides was known. Storing information or how DNA was copied was not known. In 1953, James Watson and Francis Crick from Cambridge, England, published a model of the structure of DNA. Okay, again, a question. If you would please take a moment to answer the question before moving on. Watson and Crick's success was making models of DNA using all available evidence. Apart from Chargaff's data, another vital piece of evidence came from X-rays by Rosalind Franklin. Her photographs suggested that DNA had a helical structure. Eventually, their idea of the molecule having two polynucleotide chains, the two chains were held by hydrogen bonds between the bases. The model showed the only way for this to occur was to have a helical shape, that double helix, and to run the strands in opposite directions, 5' to 3'. Referring to the sugars. That's that anti-parallel structure. At the 5' end, there's a phosphate, and the 3' end, there's a sugar. The 5' and 3' are the carbon numbers on the atom of the sugar. DNA has the following features. It is made of two polynucleotide chains. Each chain is a right-handed helix. The two chains coil around each other to form a double helix. The chains run in opposite directions. They are said to be anti-parallel. Each chain has a sugar phosphate backbone with bases projecting at right angles. The bases in one chain are attracted to the bases of the other chain by hydrogen bonding between each base. Adenine pairs with thymine and guanine pairs with cytosine are considered complementary base pairs. Adenine links with thymine with two hydrogen bonds, while guanine is going to link or bond with the cytosine with 3 hydrogen bonds. Adenine and guanine are the purines. Thymine and cytosine are pyrimidines. A purine always pairs with a pyrimidine. Purines are 2 rings. Pyrimidines are only 1 ring. The distance between the backbones is always going to be 3 rings wide. A complete turn of the double helix takes place every 10 base pairs. The 2 strands are complementary. Watson and Crick noticed how structure explains how the DNA stores information and how it copies itself. The nitrogen base sequence acts as a coded message. Taking a look at the structure of RNA, RNA molecule is a single polynucleotide strand. There's 3 types of RNA. You have messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA remains as an unfolded strand. Transfer RNA and ribosomal RNA fold up into complex structures. DNA replication is considered a semi-conservative replication. Semi-conservative meaning that half the original DNA molecule is kept in each of the new molecules. So when you look at this, you know that you have 1 old strand in each of the new molecules of DNA. That's what they mean by semi-conservative. Replication takes place during the S phase or synthesis phase of interphase, which is part of the cell cycle. In the 1950s, it still was unknown how DNA was replicated. There were 3 different possibilities. The conservative replication, where 1 complete new helix was made from an existing one. The semi-conservative replication, where each helix contained 1 new strand and 1 old strand. A dispersive replication, where each new molecule was made from bits and pieces of a new and an old molecule. This is showing again the 3 different suggestions. Semi-conservative, where you have an original strand as well as a new strand. So you had the templates were made. The conservative is where you had a segment of DNA and a new segment was completely made. The dispersive is when you had a segment of DNA and then new segments were formed, and so each strand of DNA was a mix of old and new. To show how DNA replicates, Melson and Stalin did an experiment where they grew bacteria in nitrogen-15. This nitrogen-15 is an isotope of nitrogen. Nitrogen normally has a mass of 14, but they grew the bacteria in a heavier version. The reason that nitrogen-15 was important is because they could take samples of it and then put it into a centrifuge that spins it really fast, and it would separate it based on its mass. And so at the start of it, they noticed they had the nitrogen-15. It was a heavier form of nitrogen, so it's all at the bottom of the centrifuge. But then each generation afterwards, they'll notice that the amount of the nitrogen-15 was going to start to decrease, and the nitrogen-14 was increasing. So where did the nitrogen-14 come from? They had to introduce nitrogen-14 into their experiment. So they started with nitrogen 15, allowed for a couple of generations, then introduced nitrogen 14 to see how the two different forms of nitrogen would impact on the DNA replication. And they noticed that each generation, the red is representing nitrogen 15. That began to decrease, and then the nitrogen 14 began to increase. So this is where it led both of the scientists to say that DNA replication was based on a semi-conservative, where you have one original and one new strand. DNA replication is controlled by enzymes. It begins with the unwinding or the separating, breaking those hydrogen bonds between the two nitrogen bases. That's what's known as the unzipping of DNA, and that's done by helicase. DNA polymerase is used for the copying process. The DNA polymerase is going to attach each of the single strands. It allows for a new nucleotide to attach one at a time, held by a hydrogen bond. The DNA polymerase can only copy from a 5' to a 3' direction. Remember, DNA polymerase copies from a 5' to 3' direction. So when we have a strand of DNA that has unzipped, that's the helicase has come through and it's unzipped it, we have our parent or original strand. Parent or original strand is representing this top one in blue, this bottom one is in red. Since our parent strand is going from a 3' to a 5', our copied strand is going to go from 5' to 3'. Remember, they're going to be anti-parallel, so if our parent is starting with 3', then our new strand, which is going to be known as the leading strand, is going from a 5' to a 3'. The bottom parent strand, it's going from 5' to 3'. Again, DNA polymerase can only go 5' to 3'. Since it's anti-parallel, this one is copying, but it's copying in the opposite direction of the unwinding. As a result, you get these little fragments that are forming that are called Okazaki fragments. The new strand that's formed, this is going to be considered a lagging strand. The DNA ligase has to finish the process by connecting all of the new nucleotides with the covalent bonds. DNA ligase finishes the process by connecting all the new nucleotides with covalent bonds. Before this, the complementary bases were held with just hydrogen bonds. DNA ligase connects neighboring nucleotides with the phosphodiester bonds to form the sugar phosphate backbone of the new DNA. Couple more questions for you to answer if you would take time to answer these before moving on. Watson and Crick realized the DNA code was the sequence of bases in the DNA. Other scientists realized the sequence of the bases must be the code for the sequences of the amino acids in the proteins. Enzymes control activities in the cells, and enzymes, remember, are proteins. Each protein has a unique sequence of amino acids which determines its structure and function. The sequence of the bases in the DNA of a cell is the code for all the proteins of that cell and organism. The code for one polypeptide is called a gene. There are 20 amino acids, but only 4 different bases in DNA to code for them. The codes are triplet codes, therefore there would be 64 possible combinations of 3 bases. Most amino acids have more than one code. By 1964, it was possible to look up any code. Another question for you, if you'd please take a moment to answer that question before moving on. Features of the genetic code. 3-letter code, meaning a triplet code. 3 bases make the code for one amino acid. TAC, meaning thymine, adenine, cytosine, is DNA code for methylene. The code is universal for all living things. The code has punctuations. 3 of the DNA triplets act as full stops in the message, which marks the end of a gene. Some triplets can act as a starting signal. The code is described as redundant or degenerate, meaning some amino acids are coded for by more than one triplet. The amino acid cytosine is coded for by ACA as well as ACG. The 3rd letter can be A or G, which are both purines, so the code is an AC purine. We will look at this more in part 2. One last question, if you would please take a moment to answer this question.